Hi everyone, welcome to the video, and today I have the extension of the previous topic. And if you recall, our previous topic was on gene. We tried to understand what is gene. We tried to understand the traditional definition or the ancient definition of the gene. We also tried to discuss the modern definition of the gene, or what is the modern concept of the gene. Anyways, I hope you all are enjoying these videos, and at the same time you are also learning something from these videos. And in this one we have started the biochemistry series. We will be covering a lot of different topics. We will be covering also different fields of the biosciences. So if you're new to the channel, then please do subscribe to the channel and become the part of our basic science series family. All right, so let's jump on to the topic. And as you can see, the layout is pretty simple. Let me explain again that you can see me at the same time. You can also see what kind of work I'm doing at the same time. I've done some work already to save you the time, because right now the attention span of the students is very low, and it is very difficult for teacher like us to engage you guys. So I can't just keep on writing, and I'm not able to edit while after the video is over and just trim down all the sections. So I've already written few things, and maybe you will be able to better understand these topics than you just read the books or watch videos. All right, so we'll start the video. Here we'll directly talk about some of the important things, like the size of the DNA molecule. Now, if you just recall, the size, when we talk about cellular size, when we talk about the size of the organism, it's very tiny. That is why we call them microorganisms, because they are in the range of micrometer, right? And when it comes to the size of the DNA, let me adjust my right notepad, because I'm not getting straight lines, and if I'm not getting straight lines, that will basically annoy me. Okay, now I think it's much better. All right, so when we talk about the size of the DNA, obviously it is going to be much smaller because it is inside the cell. It's going to be much smaller, and it's in the range of nanometers. All right, so when we talk about cells, when we talk about organisms, the size of DNA, here what I have written is basically it's when it is compacted in chromosome, the length of that DNA molecule is more, it's not less than the size of the cell or the virus, because it's a linear molecule, right? It's a linear molecule, and you can stretch it out. It's going to be a thread-like molecule, and it's going to be very long. Now we'll try to understand how much long it will be, right? So I have written some of the examples here. On the left side I have T7. Those who are not aware of T7 is the bacteriophage, and this is also another bacteriophage. And then we have T4, which is another family of the bacteriophage. So T4, T7, they are the name of the bacteriophages. They are E. coli phages, so they basically survive on E. coli. And what are phages? Phages are the virus particle that feeds on the bacteria. They are also known as bacteriophages. So right now you just try to understand these are bacterial viruses. Now, if we talk about the size of the DNA that is inside those virus particles, and then we'll compare the nanometer length of that DNA, and compare the length of the virus or the bacteriophage. In this case, you will be surprised by knowing all those numbers. Obviously, I'm not going to remember them, so I have to look into my notes. So for T7, just for T7, it's 39,936 BP, base pair. So that's the unit. How many base pairs you have? 39,936 base pairs. And then if you talk about lambda, it is a little bit larger than the T7. That is interesting. So 48,502 base pair, and for T4, 168,889. So which one is the largest? This one is the largest, T4, as compared to the T7. Okay, on this second place you have lambda, and third place you have, I should write it down. So this is the largest. This is, should I like at least write L in this case? So largest. This is the medium sized, and then smaller size. Okay, so when we talk about the nanometer length of this DNA, so the length, before writing this, I will move on to the length of the virus particle, or the, in this case, bacteriophage. So length of the bacteriophage is 78 nanometer. They are virus particles, so obviously they are going to be much smaller than the cell, so they are in the nanometer range. 78 nanometer, and 190 nanometer is for the lambda, and then 2010 for the T4 phage. So you can see these are important things. You need to remember these numbers, you need to remember the pattern of these numbers, and then if you move on to the length in nanometers, you will be surprised how large the DNA molecule can be. In this case, it's 14,377 nanometer. So how much the length of this particular DNA that is there inside the virus particle, which is approximately 78 nanometer. Okay, we'll come on to the topic where we'll discuss how it is possible. So 17460 for this one, and then 60800. Obviously it will be related, because more the base pair you have, longer the size of the DNA molecule will be. So you can see, but how it is possible putting all the DNA inside this small virus particle? How it is possible? It is possible by compacting the DNA. So the DNA is tightly packed, especially in case of cells, you have chromosomes. So DNA is basically compacted into these compact structures called chromosomes. In case of virus, the DNA is also compact in the compact form. That is why it's been packed. How it has been packed is also another complex research area where people are working to identify how the DNA is being packed. What is the mechanism of packing of the DNA in case of virus particle? So see, these are some interesting numbers. So we talked about size of the DNA specifically related to the bacteriophages. Okay, now this part is done. So you can see I have like these dotted lines, and these dotted lines basically will contain a specific specific topic, and then slowly we'll move on to the next topic, right? So I have multiple dotted lines. Moving on to the next, and you can see I have these filling the blanks, so I'll try to fill them while I'm writing. So it will be an interesting way to actually make you understand what are different points. Okay, now moving on to the viruses. So since we talked about viruses, they are not free living. So this is important point to remember, that they are not free living. Okay, they need a host. They are also known as parasites. They are obligatory parasites. So they need a host cell in this case to survive. So viruses, they basically will not replicate by themselves. They need a host machinery. So that is why we call mammalian viruses, plant viruses, animal viruses, bacterial viruses, because they have their specific host. In this case, you can have DNA or RNA as their genetic material, and the protein, right? Protein basically is the material by which the coat of the virus particle is made up of. If you see the structure of a typical bacteriophage, what you will have is icosahedral shaped capsid. So this is made up of protein, and inside that you can have genetic material packed like this, and then obviously you will have collar, and then you can have tail fibers and so on, right? So this is the typical structure, and I will not go into the detail of the structure, but maybe in future we'll encounter this topic again and we'll try to understand that. Okay, so next important point regarding the HIV virus, because we know that HIV is human immunodeficiency virus, one of the important and deadly disease, and we don't have any specific therapy against this particular virus, and people are dying because of this deadly disease. Okay, maybe at one point some of you will able to do research on HIV, and maybe you will try to find a cure against this deadly disease. Okay, HIV. I hope you like this illustration. HIV. So what HIV has is basically how long? 900 nucleotide long, and I'll pick another color. I don't think so. It's a single stranded RNA. That is the genetic material of this particular virus. So this was a little bit information regarding the virus DNA. So in addition to the basic concepts, we talked about the bacteriophages and the size of their genomes, and then we moved on to understanding the HIV virus, not in detail, but just the size of that, and some of the interesting facts. Okay. All right, what are those interesting facts? Let's see. Viral DNA, right? So some of the interesting facts that I've written here can have replicative forms. So are replicative forms means when it is replicating, it can acquire different types of different forms, rather than just a single strand DNA. So linear DNA can become circular, single strand DNA can become double stranded. So these things can happen. So those are the replicative forms of the DNA or the genome, viral genome. So in this case, linear DNA can become circular, right? So let me write it down, circular. And the single-stranded DNA can become double-stranded DNA molecule. That can happen. So these are the replicative form of the viral DNA. Now moving on to the next one, it's E. coli. So now you can see E. coli when we talked about the lambda phage in our previous video. Obviously now when we compare the phage DNA, compare the phage DNA with the bacterial DNA, what will happen? The bacterial DNA or genome or nucleoid will be larger. So how much larger? In terms of the times, with how bigger the bacterial DNA is, 100 times. So 100 times approximately, right? 100 times larger than the lambda phage DNA. Lambda phage we are considering because it's average sized DNA, viral DNA, as compared to the T4 that we have seen is the largest, and the other one, which is T7, is a little bit smaller, and in between you have lambda. That is why we are trying to compare. So approximately 100 times. Next, the DNA. This is interesting. So DNA itself is much longer than the E. coli cell itself. So suppose this is the E. coli cell, and how much times? It's 850 times longer than the cell itself, and which is, you will be surprised to know this number, 1.7 millimeter. Millimeter, we are talking about mm here, not nanometers, not micrometers, it's millimeter. So 1.7 millimeter of bacterial DNA, when we talk about E. coli, it will be 850 times larger than the cell itself. So you can understand how, again, how compactly it has been arranged inside the cell. Next, E. coli DNA. How many base pairs are there? So this number, I think it's better if you remember this numbers, which is 4639 and 675 base pair. These are the base pairs in E. coli DNA. Okay. So these are some of the interesting facts regarding the bacterial, especially the E. coli DNA. Now moving on to some other things, then we knew that what is nucleotide, right? There are some other circular DNA molecules that are present inside the bacteria. They are known as plasmids. Plasmids. Plasmids are nothing but the circular piece of DNA, smaller. They are much smaller than the typical nucleoid. They are very, very small. So they are basically few thousand base pair in length, or maybe some of the plasmids, they can have more than or equal to 10,000 base pair as well, right? So this is an important number regarding the plasmid DNA. Now moving on to the example where you can find plasmids. So obviously you can find in bacteria, you can find them in fungi, other fungi like yeast, you can find them in yeast, and etc. Right? So not in all fungi, but specifically example I've given is the yeast. Next, plasmid carry genes. Now, what is the need of the plasmids? Right? So plasmids are basically not that useful for the bacteria, but they can sometimes carry some useful characters like antibiotic resistance genes. Okay, so it looks like I'm coming up with very weird term, antibiotic resistant gene. You don't write it like ARG. I think I should write it down. This is not conventional. So antibiotic resistance genes, right? Genes. So these are responsible for the development of resistance against the antibiotics. So as we know that bacteria, they are susceptible to many small molecules. These small molecules are basically known as antibiotics. So antibiotics are able to attack the bacteria, and then it can basically destroy the bacteria by various mechanisms. We'll try to understand those. And if bacteria acquires a resistant plasmid, or a plasmid that can produce an enzyme that can degrade basically the typical antibiotic here, in this case, what will happen? It will acquire that trait, and the bacteria will become resistant. Resistant means now antibiotic is no longer effective. So that can happen with various other mechanisms also, but one of the important mechanism is acquisition of the plasmids that contains the gene that can neutralize the antibiotics. So a lot of research is going into this area to find out how this happens, what are the factors responsible for the development of antibiotic resistance, and how bacteria actually acquire those plasmids under the antibiotic pressure. Anyways, okay, so that concludes this video. I really hope that video was helpful for you to understand some of the important things like bacteriophages, what is the number as far as their DNA is concerned, how many base pairs are there, what are plasmids, what are the roles of plasmids, what is the overall function of the nucleoid of the bacteria. We also talked about some of the numbers, and then then we also talked about significance of the plasmid. Okay, so I'll just give rest to this video right here, and then we'll move on to the another one after this, and we'll talk about the eukaryotic DNA, some of the points regarding the eukaryotic DNA as well, right? So you will see slowly we are digging more and more detail into the bacterial and as well as the eukaryotic DNA. We'll try to understand other processes. That's the main objective of the video. I hope that you will find these videos helpful, and later on you will become a kind of you will be able to understand the process, how I teach. You will be able to understand how I'm explaining things, but you have to stay and watch the videos one by one so that you can better understand all these videos and also understand the concept. All right, okay, so let's just stop the video here, and I'll meet you in the next video. Till then, take care.